Welcome to Simplified UPSC by PPP, where we listen, learn, and grow together. Detailed lecture notes in English and Hindi, plus practice questions for prelims and mains, only on our Telegram channel. Link in description. Like and subscribe for more.
Welcome back to the deep dive. Our mission today is pretty laser-focused, mastering the core concepts of NCERT Class 12 Biology, Chapter 6, the molecular basis of inheritance.
Right.
And if you're prepping for the UPSC or, just want to be genuinely well-informed on the blueprint of life, this deep dive is your necessary shortcut. We're cutting through all that dense text to find the insights that really matter.
Exactly. We're going right to the molecular level. We're moving beyond just definitions to understand, well, everything. From how we even figured out DNA was the blueprint to the elegant rules of the double helix and even the complex machinery that switches genes on and off.
We're really trying to synthesize this to give you that strategic advantage.
Mm-hmm.
Now, before we dive into the amazing history of this field, we have to pause for a moment. We just need to humbly ask you, the listener, to support this mission.
It really helps.
It does. If you find this content valuable, please consider subscribing to our channel, Simplified UPSC by PPP, on YouTube, liking this audio, sharing your comments. It all helps us reach more learners like you.
And on that note, a huge shout out to all of our existing members. Your support is absolutely vital. It's what enables us to keep bringing you these super focused UPSC relevant deep dives. If you are capable, please consider joining the paid membership on YouTube. It helps us keep this whole thing going. Thank you.
Okay, let's unpack this. So, before we even get to the double helix, scientists didn't know what the genetic material was. For a long time, protein was actually the leading candidate. Why was that?
Well, it really came down to a perception of complexity. Proteins are made of 20 different amino acids, which allows for, this incredible diversity in structure and function.
So they seemed more capable.
Exactly. DNA, by contrast, only has 4 nitrogen bases. It just seemed too simple. Scientists thought, how could something so basic hold the instructions for a whole organism?
Mm-hmm.
That bias meant the early proof was not immediately accepted.
And that journey to proving it was DNA, that starts with the transforming principle, right?
Right. We start with Frederick Griffith's experiments in 1928.
Right.
He was using Streptococcus pneumonia, the bacteria that causes pneumonia.
Yeah.
He had 2 strains.
S and R.
Yep. The S strain S for smooth had this protective coat, which made it virulent. It killed the mice.
Mm-hmm.
The R strain R for rough had no coat, so it was non-virulent. The mice lived.
The controls were straightforward, but then came the mix.
Correct. When Griffith injected mice with heat-killed S strain, which should be totally harmless, and mixed it with living, also harmless R strain, the mice died.
Which is just wild.
It's astonishing. And he recovered living virulent S bacteria from them. So the harmless R bacteria had somehow been transformed into the deadly S type. Some material had been transferred.
But he didn't know what that material was.
He had no idea. He just proved the transfer happened.
And here's where it gets really interesting, especially for exams. Proving transfer is one thing, but identifying the biochemical nature of that substance, that's the next level.
That definitive work was done by Avery, McLeod, and McCarty. Over about a decade, from 1933 to 44, they took Griffith's heat-killed S cells and purified everything.
Yeah.
The proteins, the DNA, the RNA.
And they tested each one separately.
They did. And they found that only the purified DNA from the S cells could transform the R cells. But the truly elegant proof was how they used enzymes.
This is the key part.
Absolutely. They needed to prove that only DNA was doing the job. So they added proteases to digest the proteins.
Transformation still happened.
Yep. They added RNases to digest the RNA.
Still happened.
Right. But when they added DNase, the enzyme that specifically destroys DNA, the transformation was completely inhibited. It stopped.
That's the smoking gun. It had to be DNA.
It was the conclusive proof. That experiment really marked the shift in thinking.
Okay, so once we finally accept DNA as the blueprint, the next question is, what does its structure tell us about how it works? The double helix solved the biggest puzzle, replication.
Yes. The key implication is its structure, this beautiful concept of complementarity. The 2 strands have specific base pairing. Adenine always pairs with thymine, A with T, and cytosine always pairs with guanine, C with G.
They're like mirror images of each other.
Exactly, like 2 sides of a zipper. If you know the sequence of one strand, you can instantly perfectly predict the other.
And that right there, that explains how genetic information gets passed down so accurately.
Precisely. The mechanism is built right into the structure. During replication, the 2 strands just unwind, and each one acts as a perfect template for a new complementary strand. This ensures the 2 new daughter DNA molecules are identical to the parent.
Okay, so from replicating the blueprint, we now have to build things with it. We move to the central dogma-making proteins.
And that starts with transcription. This is the first step, where you copy the genetic info from one DNA strand into a messenger RNA molecule.
Still using that complementarity principle.
It still governs everything, with one tiny change. In RNA, adenosine pairs with uracil, or U, instead of thymine.
But then comes the real conceptual jump. You're moving from a language of nucleotides to a language of amino acids to build proteins. It's like switching alphabets entirely.
It's a huge switch.
Yeah.
And that's why the genetic code was such a puzzle. You can't just match one base to one amino acid. You only have 4 bases, A, U, C, G, but you need to code for 20 different amino acids.
So one-to-one code is impossible.
Right. A 1-base code gives you 4 options. A 2-base code only gives you 16. Not enough.
So it has to be a code made of groups of letters.
Precisely.
Yeah.
It led to the discovery that the code is read in groups of 3 bases, which we call codons. A triplet code gives you 64 possible combinations, which is more than enough for the 20 amino acids.
And that brings us to frame shift mutations, which is a great way to understand this.
Yes, the source's analogy is perfect. Imagine the sentence, Ram has red cap. The reading frame is set in 3s.
Ram has red cap K. Okay.
Now, if you insert or delete just one letter, say you insert B before R in red, the whole message downstream gets scrambled. You get Ram has B, D, C, A, P. It's nonsense.
That's a frame shift mutation. It just ruins everything after the arrow.
Everything. The entire protein becomes gibberish.
Right.
But, and this is the crucial part, what if you insert a full set of 3 letters?
A whole codon.
Right. Let's insert big. The sentence becomes, Ram has big red cap. The frame is sort of reset after the insertion, and the rest of the message is preserved. This experiment was key to proving the code is read in triplets.
So non-triplet insertions are just way more dangerous.
Exponentially more dangerous.
Okay, let's pivot to efficiency. A cell can't just be transcribing and translating all the time. It's incredibly extensive energetically, so it has to be regulated.
And in bacteria, the master class in this regulation is the Lac Operon. An operon is basically a functional unit of DNA where a group of genes are all controlled by a single switch.
Let's break down the Lac Operon itself. What are the key parts?
You've got the structural genes Z, Y, and A that make the enzymes to break down lactose. Then you have the control elements. There's a promoter, where the RNA polymerase binds to start transcription. And then there's the operator.
The operator is the switch.
It's the on-off switch. And finally, you have the regulatory gene, the I$ gene, which stands for inhibitor.
And what does that inhibitor gene do?
The I$ gene codes for something called the repressor protein. And crucially, this protein is made constitutively.
Meaning all the time.
All the time. Constantly. In the absence of lactose, this repressor protein binds directly to the operator region. It physically blocks the RNA polymerase. So the system is naturally switched off.
So how does it get switched on when lactose shows up?
Lactose itself acts as the inducer. When lactose enters the cell, it binds to that repressor protein. That changes the repressor's shape so it can no longer bind to the operator.
But the block is removed.
The block is removed. The operator is now free. RNA polymerase can get to the genes, and transcription begins. The cell starts making the enzymes it needs to digest the lactose that's right there.
So it's the substrate itself that triggers the production of the enzyme needed to break it down. That is incredibly efficient.
It's the ultimate feedback loop. The cell only makes the enzyme when its food source is physically present. And, this gives us a key insight. When this kind of take control fails, the consequences can be catastrophic.
That idea of control on a massive scale brings us to the Human Genome Project, or HGP.
The mega project, as it was called. It was launched in 1990 to sequence the entire human genome. We're talking about roughly 3.1 billion base pairs. It's just an unprecedented global effort.
So when we talk about the key findings, the salient features of the HGP, what are the big takeaways?
Well, first, the sheer scale, 364.7 million base pairs. But second, and this was a huge surprise, the gene count. Early estimates were like 80,000 genes, maybe more.
And what was the actual number?
The HGP found it's much lower, closer to 20,000 or 25,000 genes. This tells us that complexity isn't just about the number of genes, but how they're regulated.
And the sizes of these genes vary wildly.
Oh, immensely. The average gene is about 3,000 bases, but then you have the largest known human gene, dystrophin, which is a monstrous 2.4 million bases long. Another huge finding. Over 98% of our genome is non-coding DNA. Figuring out what all that junk DNA does is still a massive area of research.
And this all leads to the basis of forensic science individual differences.
Right. The HGP showed that we're all 99.9% genetically identical. But that tiny 0.1% difference, which we call polymorphism, is what makes every single one of us unique.
So since sequencing all 3 billion base pairs for every person is, that's just daunting and expensive, what practical tool came out of understanding this polymorphism?
That would be DNA fingerprinting. It's a much faster, cheaper way to compare DNA sequences. It works by exploiting the polymorphism in highly repetitive parts of our DNA.
These are the VNTRs.
Exactly. Variable number tandem repeats. These are short DNA sequences repeated over and over again. And the key thing is, the number of repeats varies dramatically from person to person.
So by analyzing those patterns,
You create a unique profile, a genetic fingerprint that's specific to an individual unless they have an identical twin.
and the application of that is just immense.
It completely transformed forensic science, paternity testing. But it's also crucial for studying genetic biodiversity, tracing human ancestry, and evolutionary biology. It's a perfect example of basic science leading to powerful, real-world technology.
So to wrap up, we've really covered the foundation of molecular biology today.
We really have, from the experimental proof of DNA to the structure, the genetic code, the elegance of the Lac Operon, and all the way up to mega projects like the HGP and tools like DNA fingerprinting.
This deep dive really gives you that critical foundation you need for everything that comes next in biology and medicine. It's paramount for intellectual curiosity and, of course, for high-stakes exams like the UPSC.
This basis is the key.
It is. So we want to wrap up by thanking you for joining us. If this deep dive helped you shortcut your study time, please take a moment to subscribe to our YouTube channel, Simplified UPSC by PPP, like this audio, and share a comment. Let us know what clicked for you today.
And again, to all our paid members, thank you. Your consistent support is truly what fuels this channel. It allows us to create this high-quality, focused content. We are so grateful.
Now, for a final provocative thought to leave you with, if gene regulation, like we saw in the Lac Operon, shows that biological success relies on this tight control based on environmental cues, what does that imply about the fragility of life? What are the profound consequences when a complex human cell loses that ability to listen to those cues and a growth switch gets stuck permanently in the on position?
Thanks for watching. Follow us on Telegram and Instagram. Don't forget to like, share, and subscribe.